the good, the bad, and the ugly. So most important slide, let me first recognize all the folks that have contributed to work I'll be talking about today, including my sadly former PhD student, Liguru, former PhD student, Andrew, former postdoc, and two wonderful undergrads who are now off to bigger and better, and my current postdoc, Chris Pershey, did a lot of this work as well. So. Okay, so outline for the talk today. First, I just want to spend just a tiny bit of time, and I showed some of these slides last year, but a tiny bit of time talking about what it is we actually want from latent variable models. I think there are many exciting use cases for LVMs in neuroscience, and as other Alex was talking about earlier today, I don't think there needs to be a kind of one LVM to rule them all approach, but I think we really need to think about what the different use cases are, and so let's discuss those. For the good, one thing I think we're getting really good at with LVMs is what I call denoising neural activity. I want to show you an example of how I think that can reveal new insights into population level computations. For the ugly, I'll just show you a little bit, one of the things we try to do in the lab is to kind of show the warts, right? Like really show, not just the good example, but like kind of how hard it is sometimes to get these things to work, and hopefully help folks get along, so that we can all kind of improve together. So ugly, LVMs are hard to fit and hard to compare. And the bad, but improving, is that I think, inferring and interpreting dynamical computations, it's really easy to be wrong. So I think this is kind of where we as a field hopefully can focus down and start to improve. Okay, so just to be sure we're all on the same page, the types of latent variables models I'll be talking about today are like when we're taking, let's just say recordings of neural population activity, and for now we can just talk about spiking, and so we're taking some high-dimensional population activity, and we're trying to find some time-varying, low-dimensional representations that summarize that activity. So, the promise of latent variable models. What do we actually want from these models? I think one fantastic use is visualization. Maybe we want to use them to relate neural activity to stimuli or the task or to behavior. Maybe we want to use them to improve the performance or robustness of brain-computer interfaces. We want to estimate the dimensionality of our data. Maybe we want to talk about the geometrical properties of population activity. We want to study computation through dynamics. We want to map coordinated activity onto cell types. We want to study communication between brain areas. Maybe we want to make comparisons across animals. I think these are there are many different ways, and many that I'm not listing here, obviously, about how we could use LVMs in our field. And these are all, I would say, like kind of different aspects of interpretability. I think interpretability is a word that often, gets tossed around a little bit, and but you could think about kind of interpretability differently in each one of these contexts. So I think the desired properties of a latent variable model have to be application dependent. So just for some examples, like if you want visualization, then what's important to you about your latent variable model? Maybe you want to maximize the variance of the data that it captures with each dimension. Maybe you want to preserve distances, so the distances in your low D space are faithful to distances in the high D space. Maybe you want axes that are aligned to task or behavioral variables to help you, visualize what's going on as the animal performs a task. Or maybe you want to maximally compress the data, and all these are different things you would want from visualization, and they would have kind of different optimal or maybe a family of approaches that could do well. One application I think a lot about is brain-computer interfaces. So here, a lot of brain-computer interfaces is not just about kind of decoding offline, but it's about control, being able to give somebody something that they can use in a closed-loop fashion. So it needs to be predictable. They need to know when I, do something, I'm not going to have it like wildly fly off, but rather it's going to be controllable, predictable. Maybe, often linear is really hard to beat for brain-computer interfaces because people can adapt to them so well. One of the huge challenges in brain-computer interfaces is being robust to non-stationarities. And as I was just getting at, like offline fitting does not equal online control in brain-computer interfaces. So if that's really your application, that's something you should be thinking about, is the gap between what we do offline with decoding and online. And one more that's really near and dear to my heart is like maybe we want to use latent variable models to interpret computation through dynamics. And we've seen so much progress in the field. It's such an exciting time for that with all the types of data that we're gathering. So what types of latent variable models do we want for that? Well, you probably want a model that somehow explicitly models dynamics. Otherwise, it's a little hard to interpret computation through dynamics. And hopefully, the dynamics model is easy to interpret. One thing that isn't often talked about is not just modeling the dynamics, but modeling the inputs to a circuit. So you probably want a model that has some sort of provision for inferring inputs. And maybe you want models that are parsimonious. That is, like, if you had a model that's 1,000 parameters versus a model that's 5 parameters and they do equally well, yeah, you probably would have an easier time interpreting the 5-parameter model. Okay, but just some takeaways from my little soapbox here is that all of these are valid uses of latent variable models. So I really hope that we don't, talk in our community like there's, one latent variable model to rule them all, because there's so many different ways you can use them. So, and I think as other Alex was getting at earlier today, when we, sorry, you do some great work too, Alex. But other Alex are just better and everything. Okay. When we talk about, the applications of latent variable models, let's make our questions and our hypotheses explicit. And I think that'll just, speed communication and let us be more concrete about what we mean. And similarly, let's choose and evaluate latent variable models in the context of their applications. Okay, I think I can turn off my soapbox now. All right, so the good. Like I said, one thing that I think latent variable models are getting really good at is denoising. So taking the relativity and removing kind of variability that we probably don't care about. So many people in the room are familiar with one of the methods that we've developed, initially with David Susillo and then advanced upon in my lab, is latent factor analysis via dynamical systems. So just to very briefly run through, the core idea is that we want to train a recurrent neural network to model population activity. So that would be this red generator network here. And we want that network for a given trial or a given window of data. We want to be able to infer an initial state of that network, and we also want to be able to infer time-varying inputs. And then we can compress, we can restrict the dimensionality of the solutions that networks finds at the output by having an optional readout layer. There's machine learning architecture to make this happen, which means adding a bunch of recurrent neural networks. We don't necessarily need to go into that. But the basic idea is that typically, this model receives bin spike counts as input, and it's optimized via log likelihood to infer single trial firing rates. You typically assuming +1. Okay, so when we first released this paper in 2018, one of the demonstrations I was really excited about was, we looked at Mark Churchland and John Cunningham's data where they showed these rotations in motor cortex. And to pull these out, they averaged across trials and they smoothed the data, and they found this beautiful rotational structure. All this data is public now, so hopefully many of you are using it. It's really fun to play with. Okay, so they found this beautiful rotational structure, but they had to average across trials and they had to smooth. And so in the LFATS paper, we showed that we could infer that same structure on individual trials. And this is like a 200, maybe 250 millisecond window. So, we're inferring these rapidly changing dynamics. This is like 2,000 trials of a monkey making reaching behaviors. So that was pretty cool to be able to replicate these results, but now do it from individual trials. And that substantially increased single trial decoding accuracy. So just to summarize, up here on the right, top right is taking those representations output by LFADs and just using like simple linear regression to decode hand kinematics, so the velocity of the monkey's hand, and we showed we could do quite a bit better by using this as a sort of denoising tool. So that's great. I think one of the things that really has kept me up at night, honestly, for years from this work, is that there's a huge gap between motor cortex and its control of the arm, which is muscle output, and these really kind of like high-level abstract behavioral variables, moment-to-moment hand velocities. So, one, what does it really mean to say we're like decoding hand velocity better? Is that, have we just kind of created a clean, high-dimensional space that we can easily regress off of, or are we actually, kind of getting toward mechanism? I would say here we probably aren't. I think it's a good denoiser. I wouldn't necessarily say that means that like motor cortex is representing the state of the hand, the velocity of the hand at any moment. And yeah, so similarly, like, does this reveal anything new? Has this taught us anything about mechanism? And I would say it probably didn't. It was very cool to be able to get that far, but I don't think it does. Okay, so this is going to be a little bit different for the workshop. I know it's COSYNE. I'm going to talk about biology for a little bit. So let's switch model systems. Completely different model system, the spinal cord. So this is pattern generation in the spinal cord is one of the oldest studied problems in neuroscience. The spinal cord is responsible for producing all the muscle activity that drives our arms and our legs. And intraneurons in the spinal cord are capable of producing patterns in the absence of upstream input. So, for example, this might be sensitive. These are videos of what's called a spinalized cat, where effectively the spine has been disconnected from upstream input from the brain, and you put it on a treadmill, and you're able to see quite a variety of patterns, like coordinating limbs differently depending on the speed of the treadmill. So walking, trotting, galloping, and stuff like that. So spinal cord it's a really cool system. It's capable of doing quite a lot. But the thing that I care about is that the output is so easy to measure. It's right there. You're one synapse away. So if we can analyze the activity of the spinal cord, we can, I think, actually kind of validate our latent variable estimates and tie them very, very closely to measurable output. Okay, so there was this recent paper in Nature last year where Runeberg showed, which I think this is probably the first time that anybody had really tried, dimensionality reduction or latent variable modeling of spinal activity. I'm not sure, but it might be. But so this was in a turtle prep where he was recording muscle activity, recording activity from the turtle's spinal intraneurons, and he showed these kind of nice rotational patterns. Very simple analysis, smoothing in PCA, but pretty exciting to be able to talk about these things in a new model system, talk about low-dimensional dynamics. So what I wanted to ask, or what we wanted to ask, is how precise is the link between intraneuron population dynamics and output? And here's a case where we can finally go directly into the circuit, model dynamics, and measure output. Okay, so the setup here, just very, very briefly, multi-electrode arrays. These recordings were done by collaborator Nick Oyong. This is a spinalized cat, multi-electrode arrays in different levels of the right hemichord while recording 7 muscles, hind limb EMG recording. So just to show you kind of first off, we were able to, I think, reproduce Rune's results on the left, which is, kind of if you smooth the activity and you apply PCA, you see these nice kind of rotational patterns. It's basically a plane. You kind of see, if you look down, you see these circular projections on the PC12 plane, but basically nothing onto PC3. But then if you apply Otto Elfman's approach that we've been developing, you start to see a lot more structure, and these trajectories are single trials. So it's pretty cool to, unsurprising, I think, that there's more to it than these kind of simple planar rotations. But, we wanted to ask, well, how precisely are we kind of capturing what the spinal cord is doing here? So one thing we thought about was a lot of the cortical literature will talk about how is it that we're able to generate movements at different speeds. So there's been some great work by Mehrdad Jassayari's lab showing that when animals are timing different intervals, you see neural activity travel at different speeds. So if it needs to time a long interval, activity will evolve more slowly. If it needs to time a short interval, activity will evolve more quickly. And from Shreya Saxena and Mark Chethan, showing that for making cycling movements at different speeds, you see kind of consistent rotational patterns, but activity would be kind of in different planes where it would travel at different speeds. So this was kind of our baseline, like, okay, really cool. We should expect to see, when an animal on an individual trial is walking at different speeds, we should be able to see the neural activity moving at different speeds. So just to give you a sense of the type of variability we see in this data, on the left is EMG activity from an extensor muscle sorted by how long the muscle was on for a particular cycle. So you can see there's a large amount of variability, like at the top, the muscle was on for almost 800 milliseconds, at the bottom it's on for about 200 milliseconds, so that's quite a range. And so we're going to take the single trial trajectories that were uncovered by LFADs without any knowledge of what the muscles were actually doing and correlate speeds to how long the muscle was on. And we see this awesome nothing. Just absolutely, 0.05. This was one of those, you had the beat head against, that's how I felt when we saw these correlations. Like, okay, we have some really nice visualization, but maybe it's really not telling us anything about what the circuit is doing. Okay, but we didn't give up, because grad students are wonderful. So what we did was take those single trial trajectories and just color them by transitions in the behavior. So when does the extensor muscle turn on, and when does it turn off? Okay, and so for people who are familiar with locomotion, that would be kind of between the stance and swing phases of locomotion. So here we're now coloring. Red is when the flexor muscle is on and the extensor is off. Blue is when the extensor is on and the flexor is off. So the only thing I want to get across here is that there's actually like a really concise organization. Like, it's not that this is kind of scattered everywhere, but it's all happening kind of at the same time, same location in our state space here. So that gave us a little bit of hope that it's not just kind of outputting noise, but it's something that's really maybe tightly correlated with what the muscle is doing. So then we did a really simple analysis where we isolated this loop where the extensor muscle is on. So going from extensor onset to extensor offset. And all we're going to do is this is just 3D like state space, about as simple, PCA on the LFADS output is about as simple a treatment of the data as we could come up with. Just put planes in various places on this loop and look at the cycles that have different lengths and just see like where do we start to see timing come out. So if we look at kind of the start of extensor onset, zero correlation with how long the muscle will be on. Keep going, zero correlation. Keep going, zero correlation. This isn't looking very good. Keep going, keep going, keep going, and then boom, right there. So what we thought that depending on how long the muscle was going to be on, we'd see these traces kind of diverging, like the long trials kind of going slowly and the short trials going quickly. But what we actually see is that everybody is kind of traveling parallel until this little region, right? Oh, sorry. I just wanted to point out the correlation here, R of 0.95. I think that's the highest correlation I've ever gotten in a real data set in my life. So I'm very, very proud of the grad student. Okay, so what this kind of showed us was there's a region, like this is all just simple analysis of a three-dimensional state space. We see millisecond level correlations with the muscle output, which the model was not, knew nothing about what was going on at the level of the muscle. So that's really exciting. And that showed us that there's kind of this region in state space that the trajectories seem to hold on to. So they're all kind of traveling together, and then depending on how long the muscle is on, they kind of pause in this region of state space. So let me just show you what that looks like, the level of video. So on the left are when the cycles when the cat is going quickly, and on the right are cycles when the cat is going slowly. And just there's a region where things pause. I'll just back up so it's synced again. You should see here, things for the slow gate cycle pause there for a while the extensor muscle is held on, whereas the fast gate cycle just go through. And I really just want to show this because, honestly, for a while, it really worried me that like when we do these kind of unsupervised modeling approaches and we pull out these beautiful trajectories that maybe they're not actually like that informative about behavior, but what we're finding is in a system where we can really closely monitor output, we are getting millisecond level correlations with behavior. And that's really exciting. So some takeaways. We can see millisecond precise correspondence between population activity and output through just unsupervised modeling. I can sleep at night, maybe, so that's a nice takeaway. There's a lot more exciting results that I think we'll get this out in the next month, but we also see kind of microvolt level correlations with magnitude of muscle activation, which is pretty amazing. Some really cool stuff here. But what I want to mention is that this is kind of enabling hypothesis generation for us. So, we kind of didn't really know, we had a hypothesis for how speed would be represented in this population. We were completely wrong. But it tells us now that there's kind of a region here in state space, and now we can go back. My collaborator has a mouse model, neuropixels recordings, we can do optogenetics. We can really see like what is driving the transitions out of this region. Is it proprioceptive input and those sorts of things. So I think this is kind of a great story for LVMs, that we can use them not just as visualization, denoising tools, but for hypothesis generation, and maybe ask me again in a couple of years and a couple more grad students, and hopefully we'll have more of this story. Okay, so I think that's the good. So I feel a lot better that denoising can reveal new insights about population level computations. Now let's talk about the ugly. LVMs are pretty hard to fit, and they're pretty hard to compare against each other. So I talked about this, mess of recurrent neural networks that we call LFADs. What I didn't really talk about is the difficulty in applying this to maybe different data sets. So data set diversity is kind of an obstacle to generality of the model. So, not every brain area is motor cortex. There are brain areas with different functions, different dynamics, different inputs, different firing properties, and we would like models that are able to handle all of these things. We also, there are recording modalities with different data types and different noise distributions. And their tasks are, obviously require different computations and have different types and different levels of structure. Oh, and another big thing that, again, we don't talk about is data set size is a big deal when you're trying to fit these models, and that can affect hyperparameters. Hyperparameters are something that we hate talking about, but it turns out that's most of grad student dissent is turning the knobs on these hyperparameters, right? So just as an example, here we trained I think this is 200 different LFADS models with random hyperparameters. And what we found was, so on the x-axis is how well we think we're doing by looking at our match to the spikes, so the negative log likelihood. On the y-axis this is from synthetic data where we knew the ground truth, and how well we were actually doing. And so you can see that we were kind of doing better for a while, and then there's kind of this overfitting that was not easy to detect. And things, we think we're optimizing negative likelihood, everything's getting better, but the models are just crashing and burning. And so, just to give you a sense of what's happening here, LFADS is an autoencoder. If you give the autoencoder enough capacity, it can find the best possible solution for modeling spiking data, which is just passing the spikes from input to output. That does not help us in any way, but it can give you a really, really good log likelihood score. So, because NLL is not a reliable metric measure of model quality, typically what we had to do was do random search and then do some sort of supervised evaluation, which is like grad students looking at PSCHs and saying, this one looks pretty good, right? Or we can do behavioral decoding, like how well can we predict, velocities or some other kind of auxiliary variable. But it turns out supervised metrics or biased measures of model quality and random searches are pretty expensive. So I'll just skip through this really quickly, but we used an approach called coordinated dropout that prevented that overfitting issue. So that was really nice. So it meant that so the blue points now are with coordinated dropout, and when we thought we were doing better, we were actually doing better. So that's nice because it means we can take the grad student out of the loop and just do automated hyperparameter search. So that's helpful. So now loss is a reliable metric of model quality. And then to make things a little bit more efficient, we use population-based training, which is train several models in parallel. So you train several models in parallel, train them for a while, and then compare models. And then, when we find that some models are not training very well, we just replace them and mutate the models that are doing well until we converge onto good solutions. And so what that meant was we were able to, in an automated way, find models that worked really well on a variety of data sets. So meaning like we could put data in, hit the button, and get models that worked well, whether we're talking about motor cortex, somatosensory cortex, DMFC, which is frontal cortex. Basic idea is like, there's a lot of mess to model fitting, and thankfully, at least, in this case, the approach was able to get around that. I'll not spend too much time on this plot, but maybe some key takeaways. So, I think demonstration and proof of principle is obviously really cool, but they're really boring but important problems like hyperparameter adjustment. And another thing is like, we were trying to test our model on many different brain areas. We don't want the field to kind of overfit to modeling like one brain area within and one behavior. Like if we really solve, center outreaching on 8 targets, how far are we going to get? Probably not very far. So I think, showing the warts, which is like what are really the hard parts of getting this model to work and trying to tackle them head on, hopefully can help, be beyond this, just enable more robust and generalizable tools for the community. And yeah, that was pretty ugly. All right, the last, very quickly, is I'll talk about the bad, but I think we're improving, is that inferring and interpreting dynamical computations, it's pretty easy to be wrong. So a couple of years ago, we released this neural latency benchmark, and I was really pleased to see that a fair number of people have been using the data to help try out their models and develop. It was a huge effort, but led by Felix and Joel, and these resources are all online, so hopefully some of you might find them useful. And one of the things that has enabled is to be able to kind of track progress in latent variable modeling. So, honestly, I think before this work, like every time somebody would develop a model, you have to pick a data set, maybe an experimentalist who has a good data set that you can use, you pick your metric for evaluating the model, and we're kind of like all, I guess, operating in isolation. So at least now we have these open data sets and some sort of standards. We can argue about what the standards should be, but at least I feel like we're communicating a little bit more as a community and being able to make head-to-head comparisons. Just here I'm showing, so on the top here is what we called co-BPS performance, so that is the ability to predict held-out neurons that the model is not seeing at inference time. And on the bottom I'm showing decoding performance, and these are for a few different methods. And just what I wanted to get across here is that I don't think decoding performance is that great of a metric. A lot of times folks will evaluate latent variable models with just decoding, and it might be useful for BCI, even though it's offline is not a great predictor of online. But just there can be a wide variability in model quality, and decoding performance could saturate pretty easily. So and that can also depend on data set. But the other point I want to make here is if you look at these models, people took a variety of approaches. So like Scott Linderman's group has S5, which is a really cool multi-layer state space model. There's transformers, like spatiotemporal transformers, discriminative models. There's kind of a lot of different ways to do this. So I think the good here is that we have open data sets, standard metrics for comparison. I think we've lowered the barrier to entry for model developers and folks interested in neural data science. And we helped convey kind of the challenges of working with population data and I think encourage people to move away from just modeling the same brain area and very simple behaviors. But the bad here is that, what does this tell us about computations in the brain? I don't think it tells us anything necessarily, right? Like, some of these are like ensembles of transformers. Some of them are models that I think could be quite interpretable, and some of them are not. And this by itself does not. I don't think kind of answers the question. So I'm going to skip over some slides that I will be talking about tomorrow. So feel free to come back in 14 hours for the other talks that I need to write. Sorry. But the key thing I wanted to get across is that we are trying to advance on this other problem as well, which is giving people tools to understand the interpretability of different models. At least one of the pieces of interpretability that I care most about, which is understanding computation through dynamics. So we're creating this new computation through dynamics benchmark. Chris will be talking about it tomorrow, where we'll have a wide variety of simulated data from different tasks and RNNs performing different computations. You saw Laura's work earlier, that's part of the benchmark. We've got RNNs controlling multi-join arm models, where I have no idea how to interpret their computations. But the goal is to kind of get this out there so folks can try fitting their data trained models on these simulated data, where we at least have a kind of ground truth for comparison. Okay. There are many open challenges in latent variable models. I don't think there's any need there's no one right metric, and there's no need for the field to overfit to a given metric. And let's just all keep working together to lower the barrier to entry. We can keep getting great contributions from the machine learning community to help us improve our latent variable models.